bit closer so that the people here feels a bit more charmed, please. Okay, good morning. We have a new session of talks today, and today it's time for people, well, in earlier stage in their career. So we have today former students of this degree. So we have today José Miguel. José Miguel is a student from the first promotion of the degree. Okay, so he finished in 2019. And he did later, well, he did the final degree project at the CRG, and later did a master in data science, in data analysis, right? And today he's doing the PhD in the BSc. Okay, so welcome, please, José Miguel.
Thank you very much. So I hope all of my talk would be basically to explain you a bit of a project that I'm working right now, so you can have a picture of an area in bioinformatics that you may end up working with. But first, I will tell you a bit my path until this point, because as he mentioned, I'm from the first batch of bachelors of bioinformatics of this degree. I graduated in 2019, and I will explain you a bit of what I did until now. So during my stay, I think it was the second year, I did an extracurricular internship at the Neurobiology of Behavior Research Group at the PRBB here. And there, I mainly worked in the wet lab, so with pipettes and stuff. But also, I started to do some analysis like ANOVA and basic statistics stuff. Then, I did my curricular internship and the final grade project at the CRG in the epitranscriptomics and RNA dynamics lab, which is also at the PRBB. And I would like to tell you a bit of what I did there, but I'll try to be quick. So basically, I worked with Oxford Nanopore Technologies, which is a recently quite new technology for sequencing. It belongs on the so-called third generation sequencing technologies, because it sequences whole molecules. Usually, in the next generation sequencing technologies, you have to chop sequences to sequence them. Here, you can sequence the whole molecule. And that was an advantage for me, because there, I use machine learning to detect RNA modifications using this new sequence technology. Then, after I finished the final grade project, I stayed in the lab because they hired me for 3 extra months. And from that time, I ended up publishing 3 papers, which is quite cool. And then, after working on real projects and real data for a while, I realized that what I liked the most and what I needed to learn to progress in my career was to do a master's in data science, to deepen my understanding in deep learning, machine learning, and data processing. And I did it at the UPC, the Universitat Politècnica de Catalunya. Then I also did an exchange there, because the master's was purely data science and no bioinformatics. So during this exchange, I was able to then apply again data science knowledge in bioinformatics. And I would recommend to do an exchange, not only because it's a great experience, but also because you can learn how they do science in another country and how they do things in another country, especially working with international people. And then I came back to Barcelona, and I did my master's thesis in the Transcriptomics and Functional Genomics Lab at the Barcelona Supercomputing Center, which is a center in which they have Mare Nostrum, which is the most powerful supercomputing in Spain. So it is really interesting to work there in bioinformatics. But the projects I started doing there is what I'm going to tell you about later, so I will not go into more detail about the project yet. And then after I finished my master's thesis, they hired me for a year to work as a research engineer, so I didn't start the PhD right after the master's thesis. And that was a great time for me because during this period of time, I decided I wanted to do a PhD, and I wanted to do a PhD in that lab. Because maybe at some point you feel pressure to do a PhD, like it's the only natural path after the degree and the master's. But there are many areas, and you don't have to be sure to do a PhD right now. I always decided at the last time, at the last possible moment. But yeah, that's what I'm doing now. During this time of research engineer, I applied for different grants to do the PhD, and then I got it, and I'm doing it now. So I will tell you a bit about one of the projects that is already finished and about to get published in 1 or 2 months. And for that, I would like to start mentioning a bit of introduction and mentioning the fact that all the cells in a human organism contain the same DNA. Yet the cells are able to differentiate into tissues with very different functions, despite the fact that all contain the same DNA. And the main reason this happens is gene expression. A gene can be expressed in a tissue and not in another, or more expressed in a tissue and less expressed in another. And this gene expression is basically quantified with number of RNA molecules. So if a gene has 10 RNA molecules, it's more expressed in this cell than in another cell in which it has only 1 RNA molecule. And that, at the end of the day, distinguishes tissue phenotype. And not only tissue phenotype, it only distinguishes diseases. For instance, the insulin gene is a gene known to be expressed in healthy individuals, but it lacks in the type 1 diabetic pancreas. So this insulin example is what we are trying to study in this project. We actually asked, my question for the project was, how do diseases affect the human transcriptome, the gene expression profile, of different tissues of a human? So for that goal, we used the GTEx dataset. Basically, it's a dataset in information of about 46 tissues, and we had a notation for 60 different clinical traits, 60 diseases, which is not common. Usually when people study diabetes, for instance, they focus either on the pancreas for the disease relevance, or blood, because it's a tissue that is easily accessible. But here we were able to study clinical traits in the context of different tissues of the human population. And not only we could study this, but we had almost 1,000 individuals, which is usually high for this kind of studies. And that allowed us to study different demographic traits like ancestry, age, sex, and BMI. So we could study the effect of the diseases in the context of different populations. So for that, I will try to be brief on the method. We basically ran linear models. Linear models, we ran 1 per gene, which we had around 26K genes, and per tissue. In these linear models, we corrected for several technical variables and batch effects known to be confounding that we always need to correct for. Then our 4 demographic traits of interest, and the different set of diseases for which we had a notation. And basically, we model, as I said before, RNA counts. So how many RNA counts are associated to 1 gene in healthy individuals, and how many RNA counts are assigned to the same gene in diseased individuals. So the output of that was a huge list of number of genes affected by each disease in each tissue. So showing all the genes that we obtained wouldn't make sense. And we decided to go for a different approach to present an overview. And this is hierarchical partitioning. Basically, we wanted to see how all of these demographic traits and clinical traits that we had affected the tissue and the variability in the tissue. So for instance, I'm showing here now the effect of only including demographic traits in this approach I'm mentioning. And we have an example on the pituitary gland and in the breast in which sex is the main driver of tissue variability. when compared to age or ancestry. This means that a young female will have a more similar pituitary gland to an old female than to a young male, because sex is the main driver of variability in the tissue. So males and females are more different than old people and young people. And this depends on the tissue. Every tissue has a different behavior. For instance, we also have adipose tissue in which body mass index is the main driver of variability as expected, and we have other tissues in which age is the main driver of variability, like the arteries or the nerve. In this case, a young female will have a more similar artery to a young male than to an old female. And I would like you to think for a second, how do you think diseases will behave in this context? Maybe diseases produces the greatest amount of variability, maybe only in certain tissues, maybe only in certain diseases, but I will go right to the answer. And it depends both on the disease and the tissue. For instance, in thyroid, Hashimoto, a disease in the thyroid, is the main driver of variability. So a person with Hashimoto will have a more different thyroid to a healthy individual than a young or an old person could have. And other examples, like pancreas and nerve in diabetes, diabetes is a great driver of variability, but it's not the main one. It also influences a lot age and ancestry. So another great thing of our data set was that we had information in different tissues, as I already mentioned, and that allowed us to check the impact of diseases across tissues, in particular diabetes, which we had information for all the tissues. And here I'm showing you the genes that are shared across tissues. So diabetes affects these genes in the same way in different tissues. And we were able to find some really well-known genes, like the ones highlighted in bold, which are not relevant. But for instance, this one was known to be affected in the adipose tissue. And here we've been able to see for the first time that it's not only affected in the adipose tissue, but also other tissues like the brain or the arteries. And then we've also been able to find new genes that were not known before to be associated with diabetes. And not only we can compare the effect of one diabetes across tissues, but also compare the similarities across diabetes. Here in this table, I'm showing in the first column the genes affected by type 1 diabetes, in the second, the genes affected by type 2 diabetes, and in the third, the overlap. So we see, for instance, that in the pancreas, type 1 diabetes is affected, but not type 2 diabetes, which is expected due to the nature of diabetes. Type 1 diabetes is the one called the juvenile diabetes, and it's autoimmune in the pancreas, and type 2 is the adult diabetes. But the only tissue in which both diabetes had the same effect was in the nerve, with a significant overlap, meaning that having either one or type 2 diabetes creates the same effect in your nerves. And it's always, in the same directionality. So the genes that increase their behavior, their expression with one diabetes also increase their expression with the other diabetes. And after doing a bit of research, we realized that this was diabetic neuropathy, a pathology arising in all diabetic individuals that arises due to having high levels of sugar in the blood. And as both diabetes at the end of the day produce that, we see commonalities. But it was never studied before in that detail. Also, I've shown you an overlap between both types of diabetes, but we can also check the commonalities between diabetes and general demographic traits. Here I'm showing you, similar to the previous table, each cell is colored when the effect of a disease overlaps with the effect of a demographic trait. And the main one is sex. This means that the genes that are usually targets of a disease are genes that are also different between different sexes. So if a male has higher expression of a given gene and a female has lower, we've seen that those genes also are affected by a disease. So this highlights the importance of including demographic traits when studying different diseases, especially for drug targets. And one commonality we see again is type 1 and type 2 diabetes in the nerve. We see a significant overlap with age, which follows the same pattern as before. Here we can see that genes that are usually upregulated, that increase their behavior, their expression with aging, are also genes affected by diabetes, which suggests that having diabetes actually ages your nerves. And here I'm showing you an example of what I mean. So here on the left, we have a facet on healthy individuals. We see that young individuals have normal expression of this gene. Older individuals have lower expression of this gene, and diabetics have even lower expression of this gene. So a type 2 diabetic that is old will have even lower expression. Then this data set we had also allowed us to study images, because we had images per all the samples. And after I took a look, I realized that in the images we could already distinguish healthy individuals with diabetic individuals in the nerves, as we see that the blobs are bigger and the white space is lower. So we decided to analyze a bit this data. For that, we extracted some basic features that I will not go into detail, but we applied machine learning approaches. We started with super vector machine, which is a basic approach, and we were able to predict with high accuracies just by inputting the images if the image is a diabetic or not, up to 85%, I think it is now. So this meant that not only we can distinguish diabetics based on their gene expression profile, but also at the image level. And we decided to use this data a bit more because in our original annotation, we only had information on whether an individual is healthy or diabetic. But using machine learning, now we are able to get a continuous variable. And if we only take diabetic individuals, this continuous variable could be a proxy of disease severity. So if an individual is predicted to be diabetic with 0.6 accuracy, well, with 0.6 variable, it means that it's not super affected. And if it has 1, it means that the diabetic neuropathy is really affected. So we could use this as a proxy of disease severity. And by doing that, and running again linear models with these variables, we found 1,000 new genes that we didn't find before in the normal approach, suggesting that this is also a good practice and a good thing to do if we want to deepen even more our understanding with this disease. But yeah, now I'm working on this. I will mention the 3 things I am working on right now. Basically, I'm trying to improve with convolutional neural networks, which is deep learning and more advanced machine learning approaches to work with images. I'm also trying to check if this happens with other tissues, and I'm working with smoking annotation. Smoking annotation, because I've worked with many different clinical traits, but I've showed you mainly the diabetes results. But also we have smoking, in which we can study the effect of what happens when someone stops smoking. And as conclusion of what I presented, I would mention basically 2 things. That including demographic traits in the models when we analyze clinical traits has allowed us to find new genes that change behavior with a clinical trait that already have different basal levels across the population. And this is important because it highlights that we need to consider demographic traits in disease analysis, especially for drug targets, as you may be giving a drug to a male because you think that the expression of their genes will be lower, but a female already has lower levels, so you could decrease even to dangerous levels. And also the multi-tissue analysis, which allowed us to see that actually the nerve is the most affected tissue by diabetes, more than pancreas or the adipose tissue as we initially thought. And we found more than 2,000 new possible target genes for this diabetic neuropathy, which is actually the strongest predictor of mortality in diabetes, yet the only complication that doesn't have a treatment. So yeah, I would like to finish acknowledging the fact that I'm not working alone, even though I'm working in a computer. I have been involved in many projects with other lab mates, and we also give feedback to one another. So it's not really a work of one person alone. And that's it. I would like to thank you for your attention, and I may take questions if you have.
Okay, thank you very much for this fantastic talk. Very nice results. I think we can be proud of producing professionals like you. So we will open a round of questions. We will open just for questions strictly related to the work, and then we will keep the questions related to the career path later together with Alba. Okay.
do you talk about several methods of studying disease, but what do you think is the most efficient to achieve something related to the transcriptomic?
The method, yeah. I'm running linear models because it's one of the simplest ones that resulted in better results. Right? And I think that's what you meant in the linear modeling step, right? Yeah, you can study transcriptomic in different types of ways, but if you have this kind of data, which is RNA counts strictly, the best thing to work is with linear models. There are some other options that are non-linear to capture non-linearities, for instance, by including H². And that's something that we will miss in the models because we are ignoring that fact that non-linearities could exist. But that's what most people do because it has been shown that has efficient results and good results, and results we could trust. Maybe if we include non-linearities, we would see more genes, but maybe we couldn't trust that much the results, although we could trust them. But yeah, I think this is the best way. Thank you.
How much time did all this research took? I mean.
Okay, yeah, that's a very good question. The modeling itself, so when I submit a model to run in our computer, which is a supercomputer, it already takes a lot. It may take like 5 hours per disease. And this is studying gene expression. I also studied alternative splicing, which is another process, and that may take up until 1 day to run. So once I tell the computer I want to run the model, I get results after 1 whole day per disease. So that's a lot. I think that if we take into account everything, this project in general took 1 year. I've been working in the lab for 1.5 years, maybe, and I've been doing some things in parallel, so I'm not only working in this project, but I would say that all of this took 1 year. Maybe half if we take into account that I've been working in 2 projects at the same time. But yeah, that's it.
Any other question? Yeah.
When you said that the expression of the genes can have like different levels, did you see any correlation between this expression and the demographic conditions?
Yes. I didn't focus on the demographic results, but I already got them. And we also had really interesting things. We've seen things like age and sex having the same directionality and the same overlap, meaning that maybe a male have a more old pancreas than females, and examples like that I didn't went into detail, but we will include those results in the paper as well. And yeah, we've seen some things. I think the most striking would be that in the arteries, I think that males have some genes more expressed that are also target genes for atherosclerosis. So that's why males tend to have more atherosclerosis. Yeah, I think that would be the most interesting thing.
Okay, so we have a last question for this section.
Hi. I wanted to know, with the smoking that you're going to study, what is the criteria you predict is going to be the most dominant?
Yeah, the thing is I already have preliminary results, so I already have a bit of an idea of what I'm going to get. The most striking thing that we've seen is that the thyroid is one of the most affected tissues. And this is novel because usually studies focus either on the lung or in blood. So we've seen that the thyroid is very, very affected. The top 1 tissue most affected is the lung, as expected, but then it's the thyroid. And yeah, I think that's one of the coolest results we will get. But
Okay. So let's thank again José Miguel for the fantastic talk.
Thank you.